So I'm going to be talking to you today about how we've been leveraging a metagenomic strategy to reveal some patterns of accessory and subgenomic chromosomal evolution. And if you'll just excuse me one moment, I want to put timer on because my timer on my laptop isn't working for some reason. There we go. Right. So why metagenomics? You know, this is a Planck conference. Why am I bringing metagenomics into this? Well, metagenomics is the study of genetic material from environmental samples. They actually do have a related problem to us. So in metagenomics, they have one data set and many genomes. So for example, I could take a swab from the table, I could take a swab from your mouth, and within that, there's going to be lots of different types of bacteria. Now with polyploid genomics, we have one data set and many subgenomes. So it's a related problem. We're also related with the size of our problem. And so with metagenomics, they need to be able to perform sequence comparison very efficiently for classification. Their data sets routinely contain thousands to millions of fragments to compare and classify. And as I'm sure you can understand, this is very computationally expensive. Now, anyone who works with plant genomics also knows that our comparative genomics is also computationally expensive because our genomes are often very large and complex. So what do they use to separate these subgenomes, the genomes, sorry. So metagenomics tends to use a combination of genomic features and data features, and they use this to separate the genomes in their data set. One example is Camber Composition, and so this is the observed Camers. And so just briefly, a Camer is just a string of length K. So for example, here we have an example of a piece of DNA. I've extracted all of the Camers of length 3 from it. And so if we did that for every sequence, we can build a table like this, where it's binary, where 1 indicates the presence of the said Camer and 0 indicates the absence. We can also do Camer frequency, and so this is just the frequency of observed Camers. So instead of just 1, 0, we're now actually counting them. And then we have sequence coverage. This is how often a sequence can appear. And so, for example, we're not talking about Camers. We could have like this one sequence that appears when it's like 256 times, another one appears 5 times. And so I have to think about whether these features are applicable for plants and what I can use. And so we can definitely use Camber frequency. We can definitely use Camber composition, but we can't really use Camber sequence coverage, because I don't expect the subgenomes to be present in differing amounts at all. So that's not really a relevant feature for us. And so when I went searching for a program that uses the Camber frequency and Camber composition features, I came across SourMash. And SourMash is a very rapid program. It employs a downsampling approach, which we call min-hashing, to sample the Camer space. It's kind of just sampling the Camers and the sequences that are given. And it generates a sequence signature, It then employs statistical strategies that approximate the similarity. And so with Camber composition, we've got the Jacquard similarity, and with Camber frequency, we have the cosine similarity. We then perform clustering of the sequences, and this produces a really nice dendrogram and similarity heat map for visualization of the sequence relationships. And that's what I'm going to be showing you lots of today. And so we wanted to determine whether subgenomes had subgenome-specific Camber frequency or composition profiles and begin to characterize those. And so we gathered some chromosome scale assemblies and we generated Camber signatures for each sequence using SourMash. And we then used the plot function to cluster the sequences and visualize the relationships based on the Camber profiles. Now, what we found is that nearly all allopolyploids cluster subgenomically very beautifully. And so what we're looking at on the screen here is a result of the SourMash analysis. And so this is wheat. Wheat is an absolute behemoth, a 17 gigabyte genome of highly repetitive and very hard to work with. But I was able to do this using a relatively small amount of computational resources. And so what we see here is we have the A subgenome in red, the B subgenome in green, and the D subgenome in orange. And what we can see on the dendrogram here is that the A and B subgenome are more closely related to each other than the D subgenome. And this makes perfect sense given that the A and B subgenomes hybridized about 500,000 years ago, and the D subgenome didn't join the party until a few thousand years ago. Now, what we can see on the right-hand side is the accompanying heat map, and this is the heat map of the sequence similarity scores from the signatures. And so what you can see in the dark blue, so if we look at the scale here, a very dark blue is a sequence similarity of 1, which means they're identical, and a kind of like light yellow, almost white, indicates a sequence similarity of 0. And so what we're seeing here is we have very high intra-subgenomic similarity, and we have very fair inter-subgenomic similarity. Now, this isn't always the case. So this is Ethiopian mustard. And what we see here is a very different pattern. While we do see 2 very beautifully separated subgenomes, we see a slightly lower intra-subgenomic similarity and almost no inter-subgenomic similarity at all. So that's already one very important comparison we can make. Now, when I switched to Camber composition, I got some variable subgenomic clustering. So for example, with wheat, this is the dendrogram we just saw. This is Camber frequency. We have beautiful subgenomic clustering. When I switched to composition, we also have subgenomic clustering. It's just a little bit harder to see because there's less sequence similarity. When I use, for example, Brassica napus, they also cluster subgenomically. However, when I switch to composition, it doesn't cluster subgenomically at all. And there's almost a kind of like homologous pattern, but sometimes no pattern at all. We can also see really cool features that are noted in the literature. So for example, here we have a cotton, and so we can see the subgenomes that are separated beautifully. But one of the things we can see here is that there is a much higher intra-subgenomic similarity with chromosome A than there is D. And this is well noted that the cottons have an unequal kind of evolution going on. But importantly for us, the A genome actually contains a much higher transposable element content, and it is the transposable elements that we believe are driving this clustering. It also may be able to pick out genome assemblies. And so this is a cotton species again, and what I found is that there were 6 chromosomes that were all in the wrong places. So 3 chromosomes, 3, 5 and 7 were in the wrong subgenome, 16, 5, 23 were in the wrong subgenome, and we did have a strange outgroup with chromosome 26. And I actually presented this at a polyploid webinar, and I ended up meeting with Justin Conover, who's actually just presented. And we had a chat because when I ran this analysis again with some cottons of the same variety, we did not get this pattern. We got beautiful subgenomic clustering. And so we kind of come to the conclusion that this might be a misassembly. And that's kind of like the sign of the times, because this is actually one of the older assemblies that I ran through. So that's a potentially useful outcome. Now, an additional thing we can do is we can cluster progenitors. And so for what we see here is wheat, but with its progenitors. So we have the A subgenome here with its progenitor Triticum murata. We have the B subgenome all by its lonesome because we don't know who its progenitor is. And we have the D subgenome over here with its progenitor. And interestingly, we actually see homologous chromosome pairing with the D subgenome in the progenitor, which makes sense given that the hybridization really was just a matter of a few thousand years ago. So we don't expect too much sequence differentiation over that time. This actually gave us a bit of a pause for thought. Can we use this to rule out progenitors? And so there was a paper recently by Lee et al. In 2022 who looked into whether Aegilops spaltoides was the subgenome progenitor of the B subgenome for wheat. And so they used different features. They used single copy orthologous genes, SNP data, and reduced represented genomic regions. And they came to the conclusion that Aegilops spaltoides is not the subgenome progenitor for the B subgenome, but is related to the potential B subgenome progenitor. I wondered if sour mash would actually give me the same result, and so I ran it. And what I found is that largely I agree. And so what I did is, again, we're looking at the wheat genome with its progenitors here. And so up here we have Triticum spaltoides and we have the B subgenome. And the one thing that draws my eyes here is that they are showing much less relationship, a much lower relationship, than we're seeing with the A subgenome and Triticum murata and the D subgenome. And so this is acting differently to the other subgenomes that we can see for wheat. But it really draws my eye when I look at Camber composition. And so what we're seeing here is the A subgenome and Triticum murata. They're forming homologous pairs and they show good similarity. The same is true for the D subgenome, and we just don't see that for Aegilops spaltoides and the B subgenome. And so I'd say provisionally these agree. There's no reason for me to say that I disagree with their findings at all. Now, sour mash is also capable of producing phylogenetically coherent dendrograms. And so this is from the same publication, and I wondered whether sour mash would be capable of mimicking it. And so what I did is I generated genome signatures, and I went through the general plot process, and I compared the two. And what I'm seeing is highest levels of similarity between the images. And so, for example, we've got the 3 outgroups, and yes, they are in slightly different positions, but they are indeed acting as outgroups. We have the Aegilops group all together, and again, I see the Aegilops group all together. We have the D subgenome group together, the A subgenome group together, the B subgenome group together, and so you can see that overall we're finding an agreement even if they're in slightly different places. My next investigation was the B subgenome, and so B chromosomes are accessory chromosomes carried by some cultivars of plant species, and I was curious as to how their Camber profile would differ. Now, their origins are unclear, but they are thought to be comprised of a genome-donated DNA and also potentially organella-donated sequences. These tend to degrade over time. In fact, in one paper, it said it's a place that sequences go to die. Now, they used to be thought of as sequences of very little importance, and so they were largely ignored. However, they have recently been shown to be transcriptionally active, and this impacts on plant phenotype. Interestingly, what we're seeing is that with maize in the presence of the B chromosome, we're getting a negative impact, and with rye, with the presence of the B chromosome, we're getting a positive impact, and so that's particularly interesting. And so what I did is I ran these through sour mash, and so what you're seeing is the maize genome and its B chromosome. Now you can see that the B chromosome is sticking out. You can see fairly low sequence similarity. However, it really is important for me to note that the B chromosome is much smaller than the other chromosomes. And so, I need to normalize for that to make sure that sequence length is not having an impact. I did this, and it's not a pretty image. I'm sorry. Once you start kind of fragmenting sequences, dendrograms tend to get a bit ugly. But I have pointed out here that the B chromosome fragments are indeed joining together. However, what I've found is that the maize B clustering is indeed Camber dependent. And so this is for K equals 7. When I get into K equals 21, the B chromosomes start to cluster in with the other chromosomes. And so we're starting to see a Camber dependent pattern. On the other hand, I found that rye actually shows stable B chromosome clustering, and it remains in this pattern throughout the Camber range. Interestingly, when I look at rye Camber composition, it shows a potential B chromosome origin. So for example, here we have one of the B genome fragments, they call it chromosome 0. And interestingly, we're seeing it in this group with 7R and 5R. And 7R is noted to have been a significant donator of genetic material to the B chromosome. And so that lines up. I can't explain why the 5 chromosome is also in that group, but I thought it was an interesting find nonetheless. So when it comes to parameters, sour mash is excellent because it really only has 3 parameters, which makes it very easy to use. Its parameters are Camber size, scale factor, and profiling type. So for example, Camber composition or frequency. The subgenomic progenitor experiments did show a fair amount of robustness for Camber size and different scale factors when I played around with them, which was very interesting. However, the phylogenetic coherence does require much further investigation. I am not a phylogenetic expert, and so it's something that does require further looking into. The B chromosome experiments also showed species-specific clustering patterns and parameter stability. Now, I only have access to 2 assembled B chromosomes, and so it would be interesting if we got more. Now, the profiling types, so Camber composition or frequency, does indeed show different relationships between the chromosomes. So in conclusion, sour mash is an efficient, rapid, and easy to implement program that has very few easy to use parameters, and it can reveal very rich subgenomic relationships. Now, such relationships have been previously described in the literature, and I think this highlights the accuracy of the approach. It also holds promise for progenitor sequence discovery and may be a promising tool for whole genome phylogenetic approaches. However, this does need more testing. This approach also showed interesting species-dependent B chromosome clustering patterns and may be capable of revealing the B chromosome origin. Again, though, this requires much more investigation when I only have a sample size of 2. With that, I would like to make a whole bunch of acknowledgements. So Montana Wheat and Barley Association for the funding, Dr. Sarah Branca for her discussion on accessory chromosomes, Dr. Titus Brown for his help extending sour mash's capabilities, Coltrane Hophan Nichols, who is MSU's supercomputer sysadmin, who without I wouldn't have any results. Also the Plant Genome Online team for their accommodations. I've been very poorly and it almost looked like I wasn't going to be able to be here today, but with their help I was. And so with that, I would love to take any questions from you.